is said to be unitary if x times 1 this is equal to x. So, for all x belongs to M. Okay. Okay, so now we have two type of notions, one is left module notion, another one is right module notion. So, then whether they are same or different. Okay. So, studying about all the left modules over all rings is same as studying all right modules over all rings. Remark, studying about all left modules over all rings is equivalent to study about all. Okay. One more remark, if R is a commutative ring, then every left R module is also a right R module and vice versa. Commutative ring, then every left R module is a right R module and vice versa. Okay, now let us prove this. It is simple only. So, let me start with a left R module. So, let M be a left R module with the structure map R cross M to M. So, let us say, so this is A and X, it is going to AX. So, for all A in R and for all X in M. Now, let us define another map from M cross R to M in the following way, as if we take any X and in M and A in R. So, this is going to let us say this is, so X dot A. So, this is defined as AX. Okay, definition I am just using this notation. Okay. So, for any A in R and for any X in M. Okay. So, let us define X dot A as AX. AX is the left R module structure scalar multiplication. So, then now our claim is M is a right R module with the above defined map. Okay. So, let us verify the three conditions. So, the first condition is, so for any X and Y in M and for any A in R. So, let us look at X plus Y A, okay, dot A. So, according to our definition, this is A times X plus Y. So, then this is AX plus AY, because M is a left R module, since M is a left R module, and this is equal to x dot A plus y dot A, according to our definition of the new scalar multiplication. Okay. In the second one, so for any x in M, A and B in R, so let us look at A plus B, okay, of course x times A plus B. So then this is dot, so then this is A plus B according to definition of the new scalar multiplication, then this is A plus B times x. So then this is Ax plus Bx. So then this we can written as x dot A plus x dot B, okay. So therefore the second condition holding, and the third one, so this is the important one, AB, so x dot AB, according to our definition this is AB of x, okay. So but our ring R is a commutative ring, so then this is we can also written as BA times x, since R is commutative, okay. A and B are elements of R, therefore AB equal to BA. So then this we can written as B of AX, okay. So B of AX, then this is AX dot B, right, according to our dot definition. So then this is x dot A dot B, okay. So therefore it satisfies the three conditions also. So therefore thus M is a right okay. So this is, now let us look at various examples. So first very example is any ring is any ring R is a right R module. Any ring R is a right R module. So how does the with the structure map? So how does the structure map is it is defined? So this is just simply R cross R to R. If you take any x and this is A, so then this is same as x A, okay. This is the structure map. And the second one we can verify that it satisfies all the conditions. So then this is fine. Suppose let S is contained in R, this be a subring. Suppose this is a subring, it's a subring of R, then R is naturally a left as well as and right, sorry, right S module, okay. So how does it is naturally a left and right R module? So this is simply we can for the left, this is a left module structure. So that means let us look at S cross R to R. If you take any, so this is A and this is X, so just go to AX, just a multiplication in, this is multiplication in R, because so for any A belongs to S, but S is a subset of R, okay, and therefore A is an element of R. Similarly X belongs to R, therefore this is left module structure. Similarly right module structure is given by, so R cross S to R, so X A going to X A. Again it is again a multiplication. Okay, in R. So, for any X belongs to R. Okay. So, welcome to the lecture 33. So, in this lecture I am going to discuss about submodules, direct sum and direct product of modules, and we will see some examples also. So, let me start. I am discussing about submodules, direct sum, direct product of modules. So, let me start with the definition of submodule. So, let R be any ring and M a left R module. A non-empty subset N of M is said to be a submodule of M. N plus is an additive subgroup of M plus. Okay. So, additive subgroup. Second condition. So, N is closed under scalar multiplication. Okay. So, that is for A belongs to R and for any X belongs to N. So, AX, the scalar multiple, this belongs to N itself. So, we have so many examples I can give. So, one first example, R is any ring and I is contained in R, any left ideal, then I is a R submodule of R, because we know that R is a left R module. Naturally the scalar multiplication is the multiplication in the ring R, and if we take any ideal left ideal, so then it is always a left R module. Okay, say left R module. So, if we take any, suppose if M is any left R module, then if we look at singleton 0, the additive identity, then this is always sitting inside M is always a submodule, means left submodule. Once I said it is a submodule in the same nature as M. So, it is a submodule of M known as 0 module. Okay. It satisfies all the conditions, singletons at 0 satisfies all the conditions of left R module, hence it is a submodule. Means submodule means that itself is a left R module, that is the meaning. So, let me go to the next definition, which is direct product of left modules. So, let me start with a ring, let R be a ring, let M comma N be left R modules. Okay. So, let us take M and N, both are left R modules. Then if you look at the Cartesian product, then the Cartesian product, which is you already know, Cartesian product of M and N, so that is M cross N denoted by this one. This can be made into left R module, with the scalar multiplication. So, the scalar multiplication, it is defined like this. So, R cross, if you take m cross n to, so m cross n, so any element A, and if you take any tuple x and y, so this is going to Ax, Ay. Okay. So, for any A in R, x in m and y in n. Okay. So, by using this one, it will make it, then it will m cross n satisfies all the conditions of the left R module and known as the direct product of m mod, direct product of m and n. Okay. And we denote by, of course, it is by m cross n. Okay. The as usual meaning. Similarly, for any family of left R modules, we can define the direct product. So, let us see how we can define more generally the direct product of a family of left R modules. Say, let us say that family is m alpha belongs to some index set I, where I is an index set. Okay. This can be finite, this can be infinite, this can be countable, this can be countable infinite also. The index set I is defined as the left R module m, so equal to, so m equal to the direct product, it is just a notation. So, m alpha belongs to I, where the scalar multiplication r cross m to m. So, how we can we define? First of all, how does the elements in m look like? So, these are like x alpha belongs to I. Okay. The infinite tuples means the tuples indexed by I. So, this is a typically an element of m. Okay. And if you take any element A in R, so if you look at A, this scalar multiple of this alpha belongs to I. So, this is defined as A x alpha belongs to I. Okay. Of course, where x alpha belongs to m alpha for every alpha belongs to I. Okay. So, means the component wise scalar multiplication. So, the each component is m alpha. So, we are component wise multiplying. So, this will satisfies all the conditions of left R module. So, therefore, m is a left R module known as the direct product of, m is called the direct product of the family m alpha of left R modules. Okay. So, this is the thing, and we can see many examples. Say for examples, if we means various index sets, suppose let us take our first index set R cross R. Okay. This is a Cartesian product of R with R. Okay. So, this is a left R module. Sometimes I can take m, this is R cross R n times, so on, n times, then this is also a left R module. Okay. This is a Cartesian product of n copies of R, and I can have infinite also, for example m. So, this is equal to So, the direct product of certain R alphas alpha belongs to, let us say, natural numbers. So, where R alpha is equal to R is any ring. Okay. So, then this is also, these are all examples of various Cartesian product of modules. So, now let us come to sum of submodules. Okay, sum of submodules. So, let M be a left R module where R is any ring. Let P and Q contained in M be, okay, two submodules of M. Then the sum of P and Q is defined as, so P plus Q, so then this is set of all x plus y such that x belongs to P and y belongs to Q. So, let us look at all the sums x plus y, set of all sums x plus y, x belongs to P and y belongs to Q. So, this is P plus Q is defined like this. So, then it is clear that this is a subset of M, right, because both P and Q are subset of M, and clearly P plus Q is a submodule of M submodule of M, okay, because it satisfies all the conditions. Scalar multiplication is same as the scalar multiplication coming from M, okay. So, therefore, it is a submodule of M, therefore, it is known as the sum of the modules P and Q, okay. So, more generally, submodule of M, then more generally the sum of a family of submodules M alpha belongs to I of M. If you look at any family of submodules, so M alpha of M is defined as the following, okay. So, and we denote summation M alpha, say alpha belongs to I, where I is some index set, okay. So, I is an index set. So, this can be finite, infinite, etc. So, then this is defined as set of all elements summation x alpha belongs to I such that this x alpha belongs to M alpha for each alpha, x alpha belongs to M alpha. So, for all alpha belongs to I and x alpha equal to 0 except finitely many alphas, except finitely many. So, then these sums are not just finite sums only, okay. What the sums we are considering, these are finite sums only, even though I is an infinite set. Why? Because x alpha equal to 0 except for finitely many alphas. So, that is basically, this is nothing but this is something like this, x alpha 1 plus x alpha 2 plus and so on plus x alpha r such that x alpha I, this belongs to M alpha I for each I less than or equal to r, okay. So, that set is same as this one, okay, and it is a, of course, everything is sitting inside M, because each M alpha is a submodule of M. Then if we take their sums, that is also a submodule of M. Therefore, the sum M alpha is also subset of M. it is a left R module. Okay, it is also a left R module with, one can check, it is not difficult. So, it satisfies all the conditions of the left R module. Okay, it is also a left R module known as the sum of the submodules M alpha. Okay, some of the submodules M alpha, alpha belongs to I. Okay. So, and also one remark, the sum of this M alpha, alpha belongs to I. So, this is the smallest submodule of M containing M alpha for every alpha belongs to I. Okay. So, this is the smallest submodule of M containing the submodules M alpha, alpha belongs to I. Okay. Let us see some examples. So, let us take ring as Z and let us take P as this is 2 ideal generated by 2 and Q as ideal generated by 6. Okay, let us take it as P as 4. So, this as 4 and Q as ideal generated by 6. So, then let us compute P plus Q. Of course, P and Q are both are P comma Q are submodules, right, left submodules in fact, okay, submodules of R. If I am not writing anything, just simply submodule means, it means that it is a left submodule, okay. So, P and Q are both left submodules of R, and let us compute what is P plus Q. P plus Q is also a submodule of Z. So, let us see what is that, that is this is ideal generated by 4 plus ideal generated by 6, okay. So, what is this? This is basically A 4 times A plus 6 times B such that A and B belongs to Z, okay. So, but what is this set? So, this is simply, this is also an ideal, it is generated by 2, okay. So, in fact, you can check that this is nothing but, let us look at one more example. So, let us take R is again Z, let us take M as Z square, Z power 2, okay. M is a left R module, okay, it is clear. Now, let us look at P. So, let us take it as P is equal to set of all x comma 0 or 2x comma 0, okay. So, let us take it as 2x comma 0 such that x belongs to Z, okay, and Q. So, this is let as 0 comma y such that y belongs to Z, okay. Both are contained in M, this is also contained in M. Check that P and Q are submodules of M. So, if we compute P plus Q, so what is that? That is simply 2x comma 0 plus 0 comma y. So, such that x and y belongs to Z. So, what is this? This is 2x comma such that x and y belongs to Z. So, one can check that this is also a submodule of M, okay. So, now let us come to intersection of submodules. So, let M alpha belongs to some index set, this be a family of submodules of M, okay, M is a left R module. So, then we take intersection M alpha, alpha belongs to I, this is also a submodule of M, okay. It satisfies all the conditions of the submodule. Therefore, intersection M alpha is also a submodule of M. So, now let us look at direct sum. Okay. So, let R be, so let us say this is P, Cartesian product P equal to M cross N is a left R module, right. The Cartesian product is, it is a left R module, as we have seen, it is just simply the Cartesian product. Is a left R module, then P contains M and N as submodules, namely M, we can M cross take it as 0, means singleton z0, that is the meaning. So, this is a set of all x comma 0. So, this belongs to P such that x belongs to M, and let us look at 0 cross N. So, then this is set of all 0 comma y, this belongs to P such that y belongs to N, okay. So, we can M and N we can view as submodules of the Cartesian product in the following way: M cross 0 and 0 cross N, okay. Then the sum of these two submodules of P is called, then the sum of these two submodules of P is called the direct sum of M and N, and denote by M direct sum N, okay, and we denote by like this. So, that is, we have M direct sum N. So, this is equal the points x comma 0 plus 0 comma y such that x belongs to M, okay, sorry, x belongs to M and y belongs to N, okay. So, what is this? This is precisely x comma y such that x belongs to M and y belongs to N. So, M direct sum means N has, this is the property we have. The sum is direct in the following sense. So, every element of M direct sum N can be, every element of M direct can be uniquely written as a sum of an element in M and an element in N, okay. So, every element of M direct sum N can be, it is uniquely written as sum of an element in M and an element in N, and so that is this one. So, So, let M and P. So, then M intersection N equal to 0 if and only if every element of M plus N, every element of M plus N can, every element Z, okay, let us call this some element Z, Z belongs to M cross N, can be written as, can be written uniquely as Z equal to x plus y, where x belongs to M and y belongs to N. Okay. So, now let us see the proof. So, let us prove this implication. So, assume M intersection N equal to 0. Okay. So, now let us take an element Z belongs to M plus N. Okay. We need to show that this we can write uniquely as a sum of an element in M and N. So, suppose Z is equal to x plus y, and this is has two sums x dash plus y dash. Okay. So, for some x and x dash in M, y and y dash in N. Okay. Suppose I have two such sums. So, then x minus x dash, this is equal to y dash minus y. Okay. So, then this belongs to M as well as N, but it is given to be 0. So, that means that x minus x dash, this is equal to y minus y dash, this is equal to 0. So, that means that y dash minus y. Okay. So, this is y. So, then this is x plus y, this is equal to x dash plus y dash. Okay. Therefore, Z can be written uniquely. Okay. Now, let us look at the reverse inclusion. Suppose Z we can write uniquely, then we need to show, so M intersection N is equal to 0. Okay. So, let me start an element Z belongs to M intersection N. Okay. So, then Z plus minus Z, if you look at this one, Z, this belongs to M, minus Z, because Z belongs to the M intersection N, Z belongs to N also, therefore, minus Z belongs to N. Therefore, if we take the sum, then this is 0, we know Z plus minus Z is 0. So, the 0 we can write 0 plus 0. Okay. By uniqueness of the sum, by uniqueness we have Z equal to 0 and minus Z is also equal to 0. So, that is Z equal to 0, but Z is an arbitrary element. So, therefore, M intersection N has to be 0.
Swayam Prabha, Digital India, Educated India.
Welcome to my course on genome editing and engineering. We are discussing about genome editing and personalized therapy. This is lecture 2 of this module number 11. Genome editing in the development of personalized therapies. The disease factors like oncogenes, cancer stem cells differ from patient to patient. So, personalized therapies appear to be a promising solution as they allow for the precise destruction or restoration of disease cause based on comprehensive genomic analysis of various patients. The 21st century has seen extraordinary advancements in customized therapy as one of the most promising strategies for various human diseases. With the use of personalized medicine, medical professionals may give and plan customized treatment for their patients based on the distinctive genes, protein markers and other components of his patient's body. The use of Ch-15 in personalized therapy. Ch-15 driven gene disruption was primarily demonstrated in 1994 when a 3-finger protein was constructed to specifically block the expression of the BCR-ABL human oncogene that was transformed into a mouse cell line. The BCR-ABL fusion gene is the pathological reason of chronic myeloid leukemia and plays a critical role in the resistance of imatinib, a kinase inhibitor that blocks the function of an abnormal protein that signals cancer cells to multiply. So, in this publication by Chu et al., the in vivo preparation by a site-specific DNA binding protein designed against an oncogene sequence was published in the journal Nature in 1994, and you can see here the CABL protein tyrosine kinase gene on chromosome 9 was translocated into the BCR gene on chromosome 22 to create a chimeric oncogene known as BCR-ABL. The Ch-15s were designed to target BCR-ABL gene and induce gene modification. So, here in this figure A, you can see targeted sequence of Ch-15 on the BCR-ABL gene. Ch-15 was designed to cut exon 1 of BCR-ABL gene, and it consisted of 4 zinc fingers and a fork endonuclease. Together the left hand and the right hand work as dimers to induce the specific DSB, the mechanism of which has been discussed in length in under the lectures of Ch-15 technologies. In this figure B, you can see the structure of PAD track Ch-15 vector. ZFP was fused to fork1 nuclease, a nuclear localization signal, and the flag tag. The expression of kanamycin resistant gene was regulated by the CMV promoter. And in figure C, you can see the sketch of the donor construct and HDR detection scheme. Cleavage of BCR-ABL gene created a substrate for HDR which may use the donor DNA fragment containing a NOT1 site as a repair template. The introduction of NOT1 site which involve 8 base pair may result in termination of translation. A custom designed JFN was used to deliver site specific double strand breaks to the telomeric portion of the mixed lineage leukemia gene breakpoint cluster region and to analyze chromosomal rearrangements related to MLL leukomogenesis via DSB error repair in the study using a human lymphoblast cell line derived from chronic myeloid leukemia patients. Using tailored Ch-15s which encourage the disruption of endogenous T cell receptors and chain genes, successful targeted modulation was also accomplished. Ch-15 treated lymphocytes proliferated with an increase in interleukin 7 and interleukin 15, but lacked the surface expression of the CD3 TCR. Another novel therapeutic Ch-15 specifically killed human T cell leukemia virus type 1 infected cells in an in vivo model of adult T cell leukemia by targeting the promoter function of long terminal repeat from its production of HER2 positive CPP coupled to mammalian mTOR specific Ch-15 rendered the mTOR locus inactive and blocked important cancer signaling pathways offering insight into the creation of innovative molecular targeted treatments for breast cancer in particular and other forms of cancer. Ch-15s have also been applied to optimize T cell mediated anti-tumor therapy. For example, glioblastoma specific cytolytic T lymphocytes can be created by introducing a chimeric TCR containing an extracellular IL-13 domain and a cytoplasmic CD3 domain into CD8 plus T cells. To do this, Reich et al. Used Ch-15s to knock out the glucocorticoid receptor in modified cytolytic T lymphocytes. As a result, despite the presence of glucocorticoid treatment, immunosuppressive, the cytolytic activity of zitokine transgenic CTLs against glioblastomas was sustained. This method has recently been shown to moving glucose transport related genes from two glycolytic tumor models, colon adenocarcinoma and glioblastoma. The application of TALEN in personalized medicine. The use of TALENs to efficiently disrupt the endogenous genes anti-F3 and CCF5 in human leukemia cells by the insertion of EJ or HDR induced alteration into a coding region revealed that TALENs could be tailored for selective endogenous gene cleavage. Precise disruptions have also been introduced into the T cell receptor alpha constant CD52 gene in allogenic T cells by TALEN induced HDR. The retroviral vector engineered TALEN expressed a chimeric antigen receptor targeting CD19 plus leukemic B cells, which helped to develop the universal CAR T cells. Recent research has also showed that the TALEN gene editing technology, which is used to knock out genes in cancer cells, including cells from prostate cancer, breast cancer and hepatocellular carcinoma, is a powerful and broadly applicable platform for investigating gene mutations at the molecular level. It has been reported that the expression of IL-6, an important factor required for tumor growth and metastasis in hepatocellular carcinoma, could be disrupted by TALENs. TALENs were also used to target human papillomavirus E7 and E6 oncoproteins, which cause malignant conversion in cervical cancer. It was observed that the editing efficiency of E7 gene in cervical cancer cells was around 10 to 12%. In addition, the cell that induced by TALEN editing was shown to be tightly associated with cell necrosis. In this figure, in A, we can see the RT-PCR analysis for the transcripts corresponding to E7 and E6 showed a significant reduction in treated group when compared to the control group. In figure B, we can see the graphs which represents expression levels of E7 and E6 in control and in the treated groups. Immunocytochemical analysis also indicated that E7 protein levels were significantly decreased in TALEN treated HIJ when compared to the control from E to G. The development of CRISPR-Cas based approaches opens up a new opportunity for customized genetic element screening, including the identification of gene and enhancers that affect many aspects of cancer growth and the subsequent development of treatments. The ultimate objective of CRISPR-Cas9 cancer treatment is to eliminate malignant mutations and replace them with normal DNA sequences. Let us discuss about the CRISPR-Cas9 application in personalized and its potential. The CRISPR-Cas9 techniques offer enormous potential for cancer detection as below. CRISPR-Cas9 based diagnostic system Sherlock, specific high sensitivity enzymatic reporter, unlocking and detector, DNA endonuclease targeted CRISPR trans reporter for cancer diagnostics, providing T cell receptor knockout, TCR-KO, chimeric antigen receptor T cells, CAR T cells, about which we have discussed at length in the earlier lecture also. Knockout of inhibitory receptors such as PD1, program cell death protein 1 and LAG3 lymphocyte activation gene 3 to promote the capability of cancer immunotherapy. Elimination of oncogenic virus like HPV and establishment of in vivo tumor models by eliciting mutations in several genes. Genome or subgenome wide CRISPR screens in identification of personalized drug targets. This is one of the key high throughput methods of assaying contributions of hundreds or thousands of genetic elements in parallel. Genome wide or subpool CRISPR screens are employed using guide RNA libraries. A guide RNA library is a collection of multiple sets of guide RNAs. Each guide RNA sets an annotated genetic element in the genome. Cas10 expression is attained either by incorporation of its expression cassette in the sgRNA backbone or by using a separate plasmid. If all the genetic elements in a genome is targeted, the screen is called as genome-wide screen. On the other hand, screens targeting only a subset of genetic elements like oncogenes, tumor suppressor genes, angiogenic genes, metastatic genes or stemness genes are known as subpool screens. The resultant plasmid pool is amplified in a bacterial system. The representation of guide RNAs in the plasmid pool or the pool of cells initially infected with lentivirus is determined by next generation sequencing. In recent years, CRISPR screens have been widely used by many to identify the genes that contribute to several hallmarks of cancer progression, including primary tumor growth, drug resistance, epithelial to mesenchymal transition, cancer stemness, metabolic adaptation and metastasis. Bulk and single cell screening can be practiced with patient derived organoids and patient derived xenografts to model patient specific responses to therapeutic regimens, metastatic heterogeneity and further identification of individualized drug targets. Even though loss of function and gain of function screens have been utilized to detect causal genetic elements of diverse cancer phenotypes, not many have strived to understand the effect of that initial heterogeneity of cell clones on the result of the screen. Many genome-wide CRISPR screens have been performed with incorporation of barcodes in the guide RNA library to trace behavior of cell clones. Guide RNA barcode combinations are produced by introducing the barcode either exterior or within the guide RNA scaffold and works as unique molecular identifiers. The CRISPR UMI technique is more suited to explore and the underlying heterogeneity of cancer phenotypes because it can statistically normalize the effect of clone dynamics on relative abundance of guide RNAs. This figure shows the workflow of the CRISPR UMI pool screen for the discovery of therapeutic targets specific to cancer patients. Bark RNA libraries are used for ex vivo patient derived organoid based or in vivo patient derived xenograph based CRISPR screens which address the issue of random clonal drift in the cell population that is being assayed to identify individualized therapeutic targets with greater statistical influence. So, we have a cancer patients from which we obtain a tumor and have a pool of cells to be screened, and there is the lentiviral transduction where we have the U6 promoter guide sequence, scaffold and barcode, and this is the guide RNA cassette or CRISPR UMI guide RNA library in all in one vector. And due to this lentiviral transduction, we get a initial cell library which may go for genomic DNA sequencing or STS, and finally there will be data analysis. And on the other hand, these initial cell library can be used for developing of patient derived organoid studies or the patient derived xenograft studies, and there is a selection pressure due to primary tumor growth, metastasis, drug resistance, and we obtain the final cell library as a result of this. And then these data along with the original initial cell data, we go for sequencing and then the data analysis. So, this will give us information on the guide RNA total count analysis and the lineage dropout analysis, from which we try to develop the individualized therapeutic target. Non-small cell lung cancer NSCLC accounts for 80% of all lung cancer and is the leading cause of cancer related deaths worldwide. Currently, immune checkpoint inhibitors targeting either programmed cell death protein 1 or PDL1 have become part of the standard treatment for late stage PDL1 expressing NSCLC with no molecular drivers. To determine whether PD1 edited autologous T cells can be a viable alternative to antibody based immunotherapy such as pembrolizumab in cancer treatment, and to examine the general safety and feasibility of CRISPR based cell therapy, Lu and colleagues conducted the first in-human trial of CRISPR edited PD-1 ablated T cells in patients with advanced NSCLC. In total, 12 patients received transfusion of edited T cells and were monitored for up to 96 weeks for treatment related AEs. In electroporated plasmids expressing Cas9 and guide RNAs targeting the second exon of the PD-1 gene into patient derived peripheral blood mononuclear cells in order to produce PD-1 ablated T cells. In a dose escalation trial, the transfected cells were grown ex vivo for 17 to 14 days before being reinfused into the patients. So, this figure shows the study designed by Lu et al. Where PBMCs were isolated from late stage NSCLC patients and then electroporated with plasmid encoding the Cas9 and gRNAs targeting the second exon of PD-1 gene. The edited T cells were expanded in vitro. There is expansion of these edited T cells for 17 to 40 days as already mentioned before being reinfused back into the patient. Treated patients were monitored for up to 96 weeks for in vivo persistence of edited T cells, treatment related AEs and disease progression. They use both targeted next generation sequencing and unbiased whole genome sequencing to determine the kind and frequency of off-target mutations in order to assess the effects of CRISPR-Cas9 off-target editing in modified T cells. Only on average 0.05% mutation rate was found in 18 possible off-target locations using high coverage NGS. In contrast, using the reduced coverage WGS technique, no mutations were found in 2086 projected off-target locations. Patients receiving infusions had up to 2 years of treatment related AE monitoring. 11 out of the 12 individuals experienced grade 1/2 adverse event. Grade 3+ AEs were noted. The 2-year follow-up period saw no treatment related dose limiting toxicities, indicating that the infusions of PD-1 edited T cells were all well tolerated. Let us now discuss about another disease in context of the personalized medicine, cardiovascular diseases. Cardiovascular disease is a severe threat to human health and the leading cause of mortality in many developed countries. A single genetic mutation or a combination of uncommon inherited heterozygous mutations is frequently connected with several distinct forms of cardiovascular disease. Clinical therapies in practice focus on reducing the disease symptoms, underlying genetic defects. Currently, the development of in vivo CVD models using gene editing technology as well as in-depth analysis of CVD pathogenic genes and their molecular processes have allowed researchers to evaluate potential role of gene therapy to control particular gene expression and enhance gene functions. Various cardiovascular research models have been generated with the use of genome editing technologies. This figure shows germline genome editing approaches and treatable cardiovascular conditions like heritable cardiomyopathies, atherosclerosis and lipid disorders, and heritable arrhythmic disorders. In normal human endothelial cells, Abrahami et al. Used CRISPR-Cas9 to efficiently ablate mesohistocompatibility complex class 2 with double gene knockout. These cells hold the ability to form vascular structures without activating allogenic CD4 plus T cells. It is promising to apply such technology in the field of allograft bioengineering, including the refinement of heart transplant. An autosomal dominant condition known as PRKAG2 cardiac syndrome is induced by a mutation in the gene which codes for the AMP activated protein kinase 2 regulatory subunit. According to a new study, PRKG2 heart syndrome and other dominant hereditary heart disorders can be affected. Cardiomyopathy is a disease of results in ventricular hypertrophy and has a propensity for arrhythmias, syncope and heart failure. A number of sarcomeric gene mutations have been linked to the condition. Mutations in MYBPC3, myosin binding protein C, account of all ACM in humans as well as a considerable number of cases of hereditary dilated and non-compaction cardiomyopathy. MYBPC3 mutation in human germ cells has been corrected successfully using CRISPR-Cas9 through microinjection of Cas9 protein with gRNA and SSODN DNA into human zygotes produced by fertilization of healthy donors oocytes with sperm from a male donor with heterozygous for MYBPC3 mutation. Recently, gene editing platforms have been developed as antiviral treatments for treating infectious diseases, either by modifying host genes needed by the virus or by targeting viral genes essential for replication. Currently, genome editing based HIV therapy involves a production of HIV resistance CD4 plus T cells by modifying infection related genes and subsequently infusing the edited cells into patients. The anti-HIV effectiveness of the Ch-15 system was initially demonstrated in preclinical test using primary human CD4 plus T cells in 2008. Ch-15 delivered by the chimeric AD5/F35 adenoviral vector disrupted almost 50% of the CCR5 alleles. A patient was functionally cured of HIV infection in 2009 after receiving allogeneic donor with homozygous CCR5 D32 allele indicating that or delta 32 allele indicating that it is possible to gain HIV resistance by imitating natural homozygous CCR5 delta 32 mutations via genome editing technology. We have discussed about these in length earlier under the lectures on Ch-15 as well as TALEN and CRISPR-Cas9. The persistent expression of high risk human papillomavirus oncogenes E6 and E7 have been linked to malignant transformation and is significantly linked to cervical cancer. The targeted mutagenesis of such high risk HPV genes using gene editing tools might be used as a possible genetic treatment to reverse cervical cancer in situ. It has been reported that Ch-15 mediated HPV 16, 18, E7 DNA disruption can directly decrease the expression of E7, which results in efficient growth inhibition and type specific apoptosis in HPV 16/18 positive cervical cancer cells in vitro. Clinical trials. Analyzing clinical trial data revealed a recent shift in the chosen genome editors as well as a sharp increase in registered trials using genome editors. The initial registered trial is from 2009, and between 2009 and 2015 less than or equal to 2 trials were registered per year, all of which focused on Ch-15s. However, in 2016 and 17 the number of trials jumped to 10 per year with a further increase to 13 trials in 2018. 2006 TALENs and Cas9 trials were registered, reflecting a change in the genome editing agent as well as a number of trials. The data may be subdivided by delivery method to see how the method chosen has shifted over time. For example, early trials entirely used adenovirus based delivery, whereas later trials also use adeno-associated virus polymer mediated plasmid delivery and electroporation. Choice of in vivo versus ex vivo delivery also has changed over time. And you can see here the data starting from 2009 to 2019, and the number of Ch-15 trials increasing and then decreasing and again increasing and decreasing and increasing, and in 2009 not a single Ch-15 trial can be reported as seen by this green colored column. And you can see for the first time the appearance of in Ch-15 TALEN and CRISPR-Cas9 technology trial in 2016, and over the years the number of the Cas9 technologies picking up somewhere in 2018 and almost similar in the other years, and while the number of TALEN trials also significant, and of course there is no any reported in the year 2018. This is the genome editing trials registered in the US clinical trials database by year wise and selected genome editor, as I have already shown you the various editing technologies, Ch-15, TALEN and Cas9. And in figure B you can see the Susan delivery method. So these are unknown. These are quite significant in a way, only small in 2019. And here you can see the use of plasmids as delivery vehicles, and the majority of the times we can see that the electroporation has been used. And then we have others like AAVs and adenoviruses, and you can see the amount or number of trials based on the basis of the delivery method. They are grouped by in vivo or ex vivo use. So this is the data for in vivo 4N48, and this is the data for ex vivo, which is much bigger when compared to in vivo. Now various delivery vectors are already shown statistically here, and this is a table where you can see electroporation, mostly Ch-15 is using them, and particularly CCR5 knockout for HIV is very prominent among here. And you can see also some trial for beta thalassemia, and many companies like Sangamo Biosciences, and then you have the universities of Pennsylvania, City Hope Medical Center doing these kind of clinical trials with a particular number and date. So this table shows you mostly about talent technology being used, and target gene and effect like TCR alpha, beta, CD52 knockout, and the PD1 and CD52 knockout. We have discussed in length about this PD1 earlier, and then most of these are falling into the ex vivo method, and you can see these are being conducted by multiple countries, UK, USA, France, Belgium, and also in certain cases, for example, here is the data for the Cas9. Again, the delivery vector is either electroporation or undefined, but here you can see then countries like Asian countries like China has also been involved in this kind of trials, including European countries like Germany and of course Australia, which is altogether in a different zone and continent. Now this is the industry sponsored CRISPR research for cell disease, anti-CD19 allogeneic and hemophilia, DMD, then you have acute myeloid leukemia, then you have hepatitis B, so on and so forth. And also certain cases, certain collaborators like Vertex and Casabia is involved with CRISPR therapeutics. Then you have Novartis and Regeneron is collaborating with the Intellia Therapeutics here, and then Juno Therapeutics and Beam Therapeutics is our collaborative partners of Editas Medicine. There are certain issues which limits in vivo clinical use of CRISPR, although both ex vivo and in vivo human clinical studies have already started, and we have also discussed many examples. There are still certain problems that may restrict the therapeutic applications of CRISPR in vivo. For example, while AAV delivery is beneficial for gene therapy as it provides the opportunity of long-term expression, this may lead to unwanted effects as the CRISPR enzyme would be expressed indefinitely. Non-viral delivery may allow temporal control of CRISPR activity, but the efficacy has traditionally been lower than viral delivery. Work with lipid nanoparticles show promise for highly efficient non-viral vectors. That specificity of delivery is also essential, as in some cases a specific organ or cell type must be targeted. Most in vivo clinical trials target tissues with direct access such as the cervix or eye or alterations in liver. As the field expands to more disease targets, the ability to target the therapy either through content or cell specific targeting will become more important. What are the limitations of personalized therapy or medicine? In terms of customized precision medicine, there is a trade-off between convenience and cost. From a perspective, the higher the degree of precision, the more complex the healthcare service will be. Gaining regulatory regulatory clearances will become more challenging as engineered precision medicine will need to be exceedingly versatile to address the demands of different patients. Only a small number of healthcare related problems may benefit from precision medicine given our present understanding of human. Greater need of professionals with knowledge in genetics and engineering compared to traditional medicine. Clinicians will need to have a basic grasp of diverse materials in order to employ devices made of modified biomaterials. From therapeutic devices, other people may not agree to disclose their static data like their genetic information. Our capacity to manage big data, which includes genetics, sensing, imaging and other action, is the foundation of precision medicine. In long run, we should be able to maintain records of people's health from conception until passing, as well as from molecular to societal level. Thank you for your patient hearing. With this, we come to an end of our discussion on personalized medicine. Welcome to my course on genome editing and engineering. In this module, we are going to discuss about ethical concerns with respect to germline gene editing. We will begin with some discussion on bioethics and biosafety. Let us now first look into the origins of the term bioethics. The term bioethics was coined by Fritz Zeher, a Protestant pastor, philosopher and educator in Hell and the Cell in 1926. Fritz published an article entitled Bioethics, a review of the ethical relationship of humans to animals and plants, and proposed a bioethical imperative in 1927, extending Kant's moral imperative to all forms of life. He evaluated contemporary physiological knowledge and the ethical issues surrounding the emergence of pluralistic In the process, Fritz reinterpreted moral obligations toward both human and non-human life forms by defining bioethics as an academic field, a guiding principle and a virtue. Let us look at some historical developments in the field. Since the time of Saraka and Sushrita, India has been blessed with a magnificent code of medical ethics. The standards for a good instructor and someone who should pursue medicine are embodied in the code of Ayurveda. Additionally, Ayurveda provides advice on how to interact with patients and their family members, as well as helpful hints for us to utilize when addressing topics like organ transplantation and brain death in the current context. If we go back to the era of World War II, heinous crimes took place on captives held in military concentration camps in Nazi Germany. During this time, defenseless individuals were forcibly subjected to medical studies with questionable scientific merit. When the victorious Allied powers enacted the International Military Tribunal out on unwilling prisoners of war. In the creation of the Nuremberg Code, a set of 10 ethical principles for human experimentation. The code aimed to protect human subjects from enduring the kind of cruelty and exploitation the prisoners endured at the concentration camps. Van Rensselaer Potter, an American scientist, introduced the idea of bioethics as global ethics in his 1971 book, Bioethics, A Bridge to the Future. With his extensive analysis, Potter presented a novel interdisciplinary idea with the aim of fusing ethics and science. He intended to create a conversation between the science of life and usable knowledge, and as a result he coined the term bioethics. Potter's bioethics united humility, responsibility, and multidisciplinary and intercultural competency. One of the landmark events in the field of bioethics and biosafety is the Asilomar Conference on Recombinant DNA, which is also considered as the beginning of bioethics in the field of biotechnology. In 1975, about 140 scientists, doctors and legislators gathered in the Asilomar Conference Center in State Beach, California, in order to debate the ethical implications of genetic engineering. Certain principles of biosafety were established at this conference with the object of preventing an accidental leakage of recombinant organisms which could affect human beings, animals, or the environment. The Asilomar Conference was a milestone for science because it was a result of self-regulation proposed by the scientists themselves. At this conference, the scientists agreed that research with recombinant birds should be outlined. Since this meeting, a lot of developments has happened in the field of bioethics, biosafety and regulation. And now many of these operating principles and guiding principles are applied to the field of gene editing, and also some new guidelines have come up in the recent years to help the field of genome editing grow and prosper in a safer way and in a more human way. Let me share with you a tweet by Nuffield Council on Bioethics. You can see here this particular tweet on January 7, 2019, and the Nuffield Council on Bioethics has presented here a beautiful graphics depicting what's on the horizon for bioethics, when they have declared to have created an infographic of developments in biology and medicine that might engage and challenge public interest and public values. Bioethics is a very big subject. As you can see from these graphics by Nuffield Council on Bioethics, ranging from human reproduction to shaping human beings, or the and society, food, farming and environment, even crime and security, and finally impacting research ethics. So I will not go into detail of this infographics, but you can see here the genome editing of gametes and embryos and other important trending topics in the field of modern biology, like whole genome sequencing of embryos and fetuses, also womb replacements, and then synthesizing entire human genomes. The idea of showing you these graphics is to show you the contextual relevance of the genome editing in respect to bioethics and how it is becoming one of the main topic of concern and discussions. So there are others like treating of biological aging and or maybe decision making in pediatric care, then microbiome resource. Many of these include the intervention of either genetic engineering or genome editing. And one of the area is the epigenetic aging, and also the epigenetics as a whole has a lot of importance from the point of view of bioethics. So let us now try to understand what actually is bioethics. Bioethics is the study of ethical, social and legal issues that arise in biomedicine and biomedical research